In this video, we will study about transcription in prokaryotes in detail. But first of all, let's understand the definition of transcription. So, transcription is the process of forming RNA from DNA. We can say the process of copying the genetic information from DNA to RNA. This is transcription. In prokaryotes, it occurs in cytoplasm because there is no defined nucleus, and in eukaryotes it occurs in nucleus, but we'll do that later. Now, first of all, let's look at central dogma, which was given by Francis Crick before we start the main process. According to Francis Crick, central dogma, which means it is a set of rules, it says that first DNA should form RNA and then RNA should form protein. The process of forming RNA from DNA is called transcription. The process of forming protein from RNA is called translation. So, this is always a direction, a one-way direction where DNA will form RNA and RNA will form proteins. This is central dogma of biology, which was given by Francis Crick. Now, let's go into the details of transcription. Now, let's talk about the enzyme which is used in transcription. So, it is DNA dependent RNA polymerase. It is called DNA dependent because this RNA polymerase needs a DNA strand to form the RNA strand. That is why it is called DNA dependent RNA polymerase. Okay, next is the conditions for transcription. So, there are two conditions for transcription. First is that the transcription occurs only in a segment of DNA. It does not occur in the entire DNA strand, but it occurs only in a small segment of DNA. So, a small segment of DNA will form RNA. One is that. Second is that only one of the strands of DNA is copied. We know that DNA is a double-stranded molecule. So, if we consider that this is a DNA double-stranded molecule and they are anti-parallel, one strand is from 3' to 5', the other one is from 5' to 3'. So, both the strands will not form RNA, only one of the strands will form RNA. Now, why is it? Because if suppose we consider that this is strand A and this is strand B, and they will form RNA, it means two different types of RNA will be formed. If there are two different types of RNA, then there will be two different types of proteins that will be formed from one DNA molecule. So, that is not at all acceptable. One reason is that two different types of proteins cannot be formed from one DNA molecule. The other is that we know that RNA is single stranded. It can make protein only if it is single stranded. So, when we look at this double stranded DNA molecule, we can see these bases. Adenine, we know that adenine pairs with thymine and cytosine pairs with guanine. In case of RNA, instead of thymine, there is uracil. So, what happens is that if we because the pairing occurs or the RNA is formed on the basis of complementarity, if we consider that the RNA is forming by RNA polymerase, adenine, so here uracil will be formed, thymine, here adenine, guanine, cytosine, like this it will be formed to whichever length. Same it will be formed on the other side also, and these two strands will be complementary. If these two strands are complementary, then RNA will become double stranded because it will join together and the protein cannot be formed. As I already told you that transcription occurs only in a segment of DNA. It doesn't occur in the entire DNA molecule. And this RNA polymerase, it always forms the strand from 5' to 3' direction. Because RNA polymerase forms in this direction 5' to 3', it means the DNA strand should be anti-parallel, that is, it should be from 3' to 5'. So, this is DNA and this is RNA, which is formed by RNA polymerase. So, always remember that the DNA strand should be of this polarity, that is 3' to 5', then RNA polymerase can form mRNA. Now, if we consider this DNA molecule, then transcription unit has three parts. First of all, the portion of DNA or we can say the DNA sequences that will form messenger RNA, that is known as structural gene. So, this is structural gene, the sequences that will on DNA which will form messenger RNA. Then what is promoter? Promoter is that DNA sequence where the transcription starts. So, this RNA polymerase, it will bind to the DNA sequence and then start transcription. So, where it binds, that portion is called promoter. And last is the terminator. By the word we can understand terminator means something which is terminated or ends. So, that DNA sequence where transcription stops, it means DNA polymerase reaches here and then stops transcription, that portion is known as terminator. Let's talk about the main process of transcription. Now, we know that RNA polymerase makes mRNA, the messenger RNA, in 5' to 3' direction. This is the messenger RNA which has to be formed. Naturally, because RNA is formed in 5' to 3' direction, the DNA strand that it needs should have the polarity of 3' to 5'. So, this is the DNA strand of this polarity. When we look at this figure, this is a double-stranded DNA molecule and we have marked all the transcription units in it. The strand which has 3' to 5' polarity, this strand is known as template strand on which mRNA will be formed. So, the strand which forms mRNA is known as template strand. The opposite strand whose polarity is 5' to 3' is known as coding strand and it will not be used during transcription, means it will not code for any mRNA. Now, we know that there are 3 regions in the transcription unit. One is the promoter. Promoter are those DNA sequences on which RNA polymerase will bind and start transcription. Structural genes are those DNA sequences complementary to that RNA strand will be formed, because this is the main area where the mRNA will be formed. And terminator is the region where transcription will terminate or end. Now, we know that RNA polymerase has to start this transcription process, but it cannot initiate the process alone. It needs an initiation factor which is known as sigma factor. This sigma factor binds to RNA polymerase. When it binds to RNA polymerase, then they attach themselves to this promoter site. As soon as they attach to the promoter site, then RNA polymerase can start transcription process. And how does it happen? Both the strands of DNA separate as soon as this sigma factor and RNA polymerase attach to DNA. Then the strands separate at that point and then sigma factor goes away and RNA polymerase starts making mRNA where the structural gene is present. So, we know this is G, then there will be cytosine, here there will be guanine, adenine, and because it is RNA, there will be uracil. So, like this, RNA polymerase will form the mRNA strand wherever the structural gene, those DNA sequences are present. As soon as it reaches the terminator end, then this RNA polymerase is attached by another factor which is known as Rho factor. This Rho factor attaches to RNA polymerase and terminates transcription. How does it terminate? It unbinds the RNA which was being attached, because here these are the bases and they are hydrogen bonds by which they are getting attached. But as soon as this Rho factor attaches to RNA polymerase, this unwinds and mRNA drops out. That small strand of mRNA that is needed is enough, and that is why it reaches the terminator and the process of transcription stops. So, sigma factor initiates the process and Rho factor terminates the process. Okay, now the last point is that it is often asked where is the promoter located or where is the terminator located. whenever we are answering to that question, we always answer it with respect to the coding strand. So promoter is located at the 5' end of the coding strand, and terminator is located at the 3' end of the coding strand. So this is one last thing. Then it's often asked, what are the steps of transcription? So there are 3 steps of transcription. First is initiation, when RNA polymerase and sigma factor attaches to the promoter region, that is initiation. Second is elongation, when sigma factor leaves RNA polymerase and RNA polymerase actually makes mRNA molecule. So when it is making that mRNA strand, that is known as elongation. And termination is when rho factor attaches to the RNA polymerase, and then finally mRNA separates, unwinds, and even RNA polymerase drops off and transcription ends. That is called termination. So also notice that the mRNA strand which is formed is complementary to the coding strand sequences. So you can see here mRNA has cytosine here, coding strand also has cytosine here. Then here it is guanine, here also there is guanine, here there is adenine, adenine, and because it is RNA, instead of thymine there is uracil. So it is exactly like the coding strand, only the difference is that instead of thymine there is uracil. So I hope you have understood transcription in prokaryotes. If you like my videos, then do subscribe to my channel and like and share. If you have any questions, then drop a comment, and thanks for watching.